Any better or is it the same?
Quite better, sir.
Okay. Okay. Is it any better now?
No, sir.
No. Okay. I will I will log out and join back. Just give me a minute.
Okay, sir. Okay, sir.
How is it? Hello?
It's fine now, sir.
Okay, good. Thanks.
Okay, sir.
So, good afternoon, everyone. We were learning about gene function. Every every gene function happens to start with interaction, right? You can take a protein or an RNA. A gene produces, say, for example, a gene, and it can produce either RNA, sometimes from RNA. I mean, if it is an mRNA, it can produce a protein. If a protein has to function, right, that means it has to modify some molecule or interact with some other molecule and form a structure. In either way, the first thing that comes to that is it has to interact with several other types of proteins, though several other types of molecules. It can be as simple as water, right? Or it can be other proteins, or it can be carbohydrates or lipids or nucleic acids. You can think of any of these, and one protein or even RNA molecule, if it is the final end gene product, they have to interact with more than one protein. Sometimes you can also have a situation like a protein interacting with another similar protein, right? But what I'm intending is that interactions are what um, are essential for a function to happen. So we can, especially those that are binding to nucleic acids, are important because they may have regulatory influences. Means that if they are binding to DNA or RNA, they may have a scope to change, and if they are regulatory, they might be having global effects in the cell. Okay, that's why that those are some of the proteins that are usually studied very well. Majority of interacting proteins that are interacting with carbohydrates and lipids are typically, mostly they are enzymes. Okay, proteins could be enzymes as well. I mean, those proteins interacting with other proteins could be enzymes like phosphatases or kinases, so on. But they can also be forming something like structures, right? Something like actin filaments or microtubules, for example. They're interacting with other molecules, and then they're forming filaments, structural elements. And you may also have these something like channels, for example. Okay. And if a protein X is interacting with other protein, you call it as homodimer if it is forming a dimer. So I hope you understand what we are trying to see. Every function is every function manifested is because of an interaction. And trying to study those interactions is very important. And that is probably what we are going to discuss today. If we are talking about RNA interacting with a protein, or a DNA molecule or other RNA molecules.
Sir,
Okay. Yes, please.
so the slide is not visible, sir.
Oh, whatever. What was I doing all along? Okay. I'm sorry again. I will repeat. Okay. So I was talking about a gene product. Okay. Assume this is the gene, and it can give end product could be RNA, like rRNA, for example, or it could be mRNA. If it is mRNA, it would have given rise to a protein. And every protein, we are trying to learn about a gene function. That means a function of either the RNA or the protein. And for all these functions, what is important is interactions, whether they are interacting with any other molecule. Every function will have to have more than one interacting partners. That's very common. Majority of them would interact with water, mostly because majority of cytoplasm is aqueous, right? And even if you have a membrane protein like this, for example, you have a protein. This part of the protein is usually hydrophobic, or maybe this outside part. But again, here it has to interact with water, and water is an important molecule, isn't it? Water determines everything in life. Firstly, because water is an aqueous solution, it is polar. It is important for dissolving. It is important for the dynamicity in the cell. Every dynamic action in the cell is dependent on water. And also, if you see, water also shapes the proteins. Proteins by themselves do not have folding capacity. It's actually the polar nature of water that makes a protein fold properly. Say, for example, you have a protein, and if it is in aqueous solution, it would have all polar groups outside on the surface. If you change this, remove water and put it into any non-polar solution, something like, say, lipid, okay, we'll just say hydrophobic solution, then all these polar molecules should be embedded inside, and outside should be hydrophobic amino acids, the groups. So every molecule in life, okay, is going to interact with water. That's no doubt. But then they also interact with many others. They can interact with proteins, carbohydrates, lipids, and nucleic acids. So these are those that are interacting with nucleic acids, either be it DNA or RNA, have important regulatory functions, because regulatory functions will influence the transcriptome and or proteome, because of which the morphology of the cell or the form or function of the cell is determined. Those that are interacting with carbohydrates, lipids, and also some of them that are in proteins, they're usually enzymes. They transform the molecules from one to the other by addition, deletion, and so on, hydrolysis, something like that. So all of metabolism is determined by the enzymes. Proteins also, those that interact with other proteins may also form structural components like actin filaments or microtubules and so on. If it is an RNA molecule, the end product gene product is RNA molecule. Again, there are possibilities. They can interact with proteins, DNA, or RNA. Again, when they're interacting with any of these, they usually have regulatory functions, but also they have functions, something like ribosome, for example. Ribosome is a mixture of RNA. I mean, it consists of RNA molecules and a lot of proteins. And together, they are catalyzing the formation of proteins, isn't it? So what we should appreciate, and if we want to find any gene function, the first thing that we need to find is what is it interacting with? What are its interacting partners? That will give you an insight about um, the consequent phenotypes at the cell level or maybe at the gene level. So, yes, we are going to today what we are going to see mostly is about these interactions. And let's see, yesterday we already started, but I'm taking a step back and going into much simpler methods, and then we will get to one hybrid system and two hybrid system. It'll be a good revision, and probably in the last class I gave you a primer on that, so we'll have a better understanding today. So there is something called as gel retardation assay. Some people, it is also called as EMSA. That is much more correct word. Electrophoretic, because the technique is based on electrophoresis. Mobility, it is about measuring or looking at the mobility, electrophoretic mobility of the bands. And what we are going to show is shift, and this is the assay. Electrophoretic mobility shift assay. So in this is an interesting technique also. So the idea would be something like this. You have a DNA molecule. One second, I'll change the color. So there is a DNA molecule, and you want to know if a protein binds to this DNA specific sequence or not. Again, every time it is not, we are always talking about specific sequence. So if you want to find that if the protein X is interacting with specific DNA or not, what we do is they would add, they would have taken tubes first, okay? I'm just setting the experiment up. In that, you would have taken equal amounts of the sequence DNA, okay? And then you would add different amounts of the protein. So here, say for example, in all the cases, there are 10 DNA molecules. I am telling so just for the understanding's sake. And we also, this is DNA molecules, okay? And here we are also going to add the protein X. How many molecules are we adding? We'll start with 0, then we will add 1 molecule, or say 2 molecules, and 5 molecules, and say 15 molecules. What would happen then? So what we will see is that in this sample, this is the electrophoretic mobility. That means based on its size, it would have run to a certain length, something like this. That's the actual mobility of the DNA without the protein. In this case, we have to also remember that the DNA is labeled. DNA is labeled. And the labeling, what do you think the labeling should be? Should it be random labeling, or should it be end labeling? I think I told you how to figure out before. Say in an experiment, if you are going to have 1,000 different types of DNA molecules, just for simple way of determining, I'm telling you. In experiment, if you have say 500 or say 10 different DNA molecules are there, different, okay? Sequence is different. Or in an experiment, there is only one type of DNA molecule, right? In these cases, we don't need a lot of labeling, because that is what we have. It's only one type, and it must have been in high quantity. So in those cases, we would use end labeling. And end labeling is done using PNK, polynucleotide kinase, and we would have used gamma P32 ATP. Remember those. Okay. And if you are going to have large quantities of DNA, something like southern blotting, where you have taken genomic DNA, fragmented it, and once you fragmented, there are several thousand types of DNA molecules, out of which you will have to find your gene of interest. That means it's a lot of DNA molecules. So in that case, the probe, okay, here it is the probe. The probe is randomly labeled. Remember that. And for that random labeling, we would have used alpha P32 CTP if you're using in vitro transcription. Okay. So I hope you are in a state where you can determine whether we should use end labeling or random labeling. So coming back to the experiment setup here. So we have taken end labeled DNA molecules, 10 molecules in each tube. And we have added protein X into each tube in the tube 1, okay? Tube 2, tube 3, and tube 4. In tube 1, we didn't add any X molecules. In the second one, we added 2. Third, we added 5. And 10th, we added 15. So this is the electrophoretic mobility of the molecule without any binding. If a DNA molecule, if these two molecules have bound to two DNA molecules, okay, their overall molecular weight is increasing. Because of the molecular weight, the DNA will have trouble moving through the gel easily. Say remaining 8 molecules are there. Assume that 2 DNA molecules are bound by these proteins because you only put 2 proteins. So they are bound, and because of which they would have traveled slower. The rest of the 8 molecules would have moved faster, and they would have come to this stage, right? Those free DNA molecules will move as if there is something like this. The rest, the ones that are bound by protein, will move much slower. So you can imagine now we increase the proportion. So you may have 5 here and 5 here. We increased even more of the protein, because of which all the DNA molecules are bound. So you see a shift, right? This should have been the DNA, but now the mobility, this should have been here, but now it is here. That's a shift. Don't ever explain it like this. This band moved to here. We don't say that. We will say that because the protein is bound to the DNA, that DNA travels slower. That is what we should say. So remember this, it's called as electrophoretic mobility shift assay, EMSA. It is also called as gel retardation assay. Because the protein is bound, the mobility is retarded. So that's what you can call. I will add a complication to this. So the complication I'm erasing, and I will ask a question, something like this. We can take a very long fragment of DNA. And then we want to narrow down where exactly is the protein binding. It's unlikely that the protein is going to coat if it is sequence specific. It is all about what we are discussing here is sequence specific binding. Our question is whether protein X binds to a particular given sequence or not. So assume we are going to bind here. It is going to bind here. Or we don't know if it is going to bind here or here. How can we determine that? We can also have logical ways of doing this. Say I will make a different DNA molecules with this much length. I'll also make a fragment like this, right? I'll call this molecule A, DNA molecules. I'm not taking whole length. I already figured that through using this assay, I figured that protein X is binding to the DNA molecule. Now I want to know where in the DNA its actual footprint is. Footprint is where it is binding. So you can design a new experiment. You make 2 fragments. This protein, DNA A will have this much sequence, and DNA B will have from here to here. Now we can do mobility shift assay again. So you'll take DNA A, and you will add protein X. You will add DNA B and protein X. You'll also add B, and you'll also add A alone. No DNA, no protein X is added. Okay. So now I will give you a question, and then you can tell me what exactly do you think is happening. This is the DNA without a binding, without any protein added. This is the bDNA, that is the mobility of the bDNA without adding any protein. But if you added the protein, it is going to, I will give you the regions here. Okay, look up on this screen. I am going to give you certain regions. I'll call this as P, this one as Q, and this one as R. My job is to determine to which region, P, Q, or R, does protein X bind to the given DNA. So for that, I'm describing. So say, for example, I got a result like this. What do you think is happening? Anybody can answer? From the given data, can you tell me where the DNA is?
Q region.
Why is that?
Because in both A and B, we got a mobility shift.
Yes.
So the common between the A and B is the Q region. So it should be in the Q region.
That's right. If it were binding only at in P, you'll only see mobility shift assay, and you'll shift in this lane 2, but not in lane 3, isn't it? Because this region bDNA doesn't have Q region, and if it were binding at R, then P would not have shown any shift. Only in the third lane you would have seen. But because it is showing mobility shift in both, what is the common region in both of them? It is the Q region. Okay, so that is also a nice logic about how people used to determine and come to closer and closer about where exactly is DNA binding to protein binding to the given DNA sequence. There are other techniques also, something like this. This is called as DNase 1 footprinting. That is, it is the exact scenario of what we are trying to discuss in the previous one. This is much more straightforward. Labeling is end labeling, because are there any other DNA molecules involved here? None. So you can, that is the crude way of trying to remember whether it is end labeling or if it is random labeling. So the goal again here is to figure out where exactly in the DNA is the protein interacting. And we use an enzyme called as DNase 1, which will digest all the DNA molecules. Okay, wherever accessible, if it is accessible, it will cleave. That's what DNase 1 does. Okay, and in this case, we are not going to allow complete digestion. Okay, if there is no DNA, nothing, DNA is completely accessible, it may cleave here, 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 and here, like this. In different molecules, you are allowing enough time only for randomly being cleaved at any one place. Something like this, not complete digestion. Okay, so you would actually have a large number of random fragments. If you have added the protein, and if the protein is binding here, say for example something like this, then that region wherever the protein is actually interacting is protected from being cleaved. Okay, and so in one, you have added the DNA without any protein. That is the control, right? So you would get, because as I said in the beginning, You are not allowing complete digestion. You are only limiting it. It's something like partial digestion. In different DNA molecules, in different locations, it will cleave. So randomly, anywhere in the DNA, you might have cleavages. But in different molecules, in different spots, it might be cleaving. But if you have added protein, that is the test, then some region of that protein, you may have in various molecules, you may have cleavage everywhere else, but not in that particular location that is protected by the protein. So that must be the footprint of the protein. Okay. So DNA is one footprinting. Footprint of the protein you're talking. Footprint of protein on the DNA. And you are determining using DNase 1. Okay. If we have, as I was describing the previous method, this one, the second DNase 1 footprinting is much better. It gives us more information and clarity, right? In the previous one, we can only narrow down to a region where protein might be binding, but we cannot say exactly which basis is actually covered, right? So that is called as DNase 1 footprinting. Now we are coming to the question that the point that we were discussing in the last class about yeast 1 hybrid system. It is also a technique to determine binding, determine if a particular DNA, if a particular protein is binding to DNA or not. This is based on GAL4. GAL4, I'm repeating what we have discussed before. GAL4 is a transcriptional factor. So it has two parts. One of it is called as binding domain, and the other one is called as activation domain. So it was supposed to bind to specific promote regulatory elements upstream of galactose or gene. And then binding domain would have interacted with these regulatory events. And through its activation domain, it would have allowed the transcription of it, right? So for transcriptional activation, you need activation domain. In which part of the gene should be activated? You need binding domain, okay? So this system was utilized by making separate, you're separating the binding domain from the activation domain, and you're adding something else. So there is no regulatory or promoter elements. They construct something like this. You have the reporter, say LAC-Z. And now you are using the bait sequence, that is the test sequence. Your question is, does protein X interact with a specific DNA or not? Specific DNA sequence, okay? So if you want to do that, the specific DNA sequence will be used as bait, will be put in here upstream of the reporter gene. And now if the protein is going to interact, so we have to find out how this protein, the protein X, interacts. So what they would have done is they would have made a hybrid protein. The hybrid protein is supposed to consist of the protein X translationally fused with activation domain of GAL4. So the question is, so you are actually making a translational fusion, right? This is the start codon, and this is the stop codon. So from here to here, it is protein X. From here to here, it is the activation domain. So you will end up getting a hybrid protein. That's why you are using one hybrid in this system. So you call it as yeast 1 hybrid system. So if protein X is interacting with this bait sequence, it will interact, and because it is translationally fused with the activation domain will allow the RNA polymerase to organize and produce the reporter, something like Z. So you'll have beta galactosidase, which will convert X-gal into a colored compound. If X does not interact with this with the bait sequence, then you will not see any blue color because beta galactosidase is not produced, because LAC-Z gene is not transcribed. Because it is not transcribed because activation domain is not there in upstream of it. Activation domain is not there because protein X would not have bound it. Okay, so whether the reporter is expressed or not can indicate if the DNA, the protein X or Y or any of them, if they bind here, the reporter will get expressed. If this protein, whichever test protein we have, it doesn't bind here, then the reporter is shut down. That's called as East 1 hybrid system. The goal of it is to find out if a specific protein binds to a specific DNA or not. Okay. Now we'll go into a complicated one, a little more complicated than this, and it's called as East 2 hybrid system. In the previous one, we have constructed one hybrid. The hybrid is about making translational fusion of the sequence protein X and the activation domain. Now, the East 2 hybrid system is for the purpose is to find protein-protein interactions. Whether protein X is interacting with protein Y to determine if the interaction is there or not. So for this purpose, making two hybrids here. The first hybrid is about generating protein X with activation domain. And the other one is you're going to have a hybrid, something like this, where you will have protein Y with binding domain. Okay, so it would have produced something like Y and binding domain. So binding is a binding domain interacts with the specific its regulatory elements, whichever are there. It is operator regions. And X is we are now interested in in the activation domain. The function is to bring in RNA polymerase or activate RNA polymerase to transcribe. Our question here is, the goal of this is to find if X and Y are interacting. So one of them you can call it as prey, that you are attaching to the activation domain is usually the prey, and the other one you are making another, that is in our situation we'll write Y and X. This hybrid, this is a hybrid protein, and we have another hybrid protein here. Then you have the regulatory region, the operator, and then you have a reporter gene, something like LAC-Z. Now, we need two things for this reporter to be expressed. The binding domain, so that it binds, it brings in the, it specifies where transcription should happen. In our case, we want the reporter to be transcribed. The second thing we need for expression is that activation domain. Activation domain should come here. Only then you will have the RNA polymerase and transcription will happen. So the goal is, if X and Y are interacting, you have the binding domain here. And because Y and X are binding, if you assume that they both have interaction, then you are bringing the activation domain to the location of this promoter, where RNA polymerase will bind and then express and then eventually give blue color. If there is no interaction between X and Y, assume that, then what would you have? You will have the binding domain, okay, and Y is here. and X and activation domain are somewhere else. There is no interaction, so there is no way of bringing the activation domain to this location, right? But if there was interaction, then activation domain as a part of the hybrid protein would have been brought to this location, and the reporter would have been expressed. Okay. So why is it called two hybrid systems? Because we are constructing two hybrid proteins. One is this, one is the prey along with activation domain. The other one is bait along with the binding domain. You are constructing two hybrid domains, two hybrid proteins, and that's why this is called as two hybrid system. And it is about trying to study if interaction happens, if there is protein-protein interaction or not. Okay. One can do many other things with this. We will probably have a chat on that in some other class. So this is Western blot. I don't think I want to discuss that. I will not discuss. Okay. I hope you know the 4 important steps. What is that? Electrophoresis. I only have one question in this, which you must have learned. Electrophoresis. Then we have blotting step. Then we have blocking step. Then we will have the primary antibody, and then we will add secondary antibody, and then visualization, detection or visualization. So electrophoresis is for electrophoretic separation of all the bands. Second one is blotting. We can use a nylon membrane and use electroblotting so that the proteins in the gel will now come onto the band. Sorry, the membrane, nylon membrane. So the nylon membrane already has high affinity for proteins. So the proteins, when you are blotting, will stick like this from the electrophoresis gel. Now, if I add antibody will stick everywhere because nylon membrane has affinity for proteins in general. So we should have, to prevent the non-specific binding, we should have coated the rest of the nylon membrane with some other protein. So usually casein is used, the milk protein, so that all the other rest of the empty spaces where the bands are not there, the protein bands are not there, they will all be covered with this casein. It's easy to obtain and it is much cheaper. One can use BSA and other things also, but that's not very economical. It's difficult. So if we now add the primary antibody, it has space only to bind with if there is an interaction between its complementary determining region and the epitope on the target protein. If that is not there, then it wouldn't bind. So non-specific binding is prevented by this step blocking. So the question again is, why should you have primary antibody and secondary antibody? Why not just the primary antibody with a tag or something? You can do that actually. But every time we want to have a conjugate a conjugate is made between antibody and this detection enzyme. Say, for example, we add horseradish peroxidase or some kind of enzyme tagged. So if I have a lab, very busy lab, and I do a lot of Western blottings, one day I want to test on X. So I get the antibodies, primary antibodies against X, and that is an expenditure, right? And I also have to get it tagged with the enzyme. That is twice the expenditure. Okay. Instead, if I only got, say, next time I want to use to do Western blotting for Y protein, then again I have to get anti-Y antibody and again get it tagged. Again, each time we are having so much of expenditure, and it takes a lot of time to get these and so on. Instead, if I have primary antibodies against X, Y, this is against X, and I can also have bi against Y, Z, or anything like that. Antibody can be bound, right? And if I have a secondary antibody, like shown here, the secondary antibody should be against the constant region. So I think all these antibodies are typically anti-IgG, IgG or so. And if you know it is from sheep or other rabbit and so on, you can get antibodies against the constant regions of these primary antibodies. Then in different Western blottings, I can use secondary antibody for this, and for this also. I can order one a big lot of secondary antibody, which is against, it is anti-constant region of the primary antibody. Okay, all the primary antibodies are same in their structure. What is actually the difference? What could be the difference? It's only in the variable region somewhere here, right? You have two light chains and two heavy chains, and there are disulfide bonds like this. The tips are usually the hypervariable region, and you also have hypervariable region, which are also going to have CDRs. This is important for interacting with the epitope. Epitope is the antibody binding binding part on the antigen. Okay, so if we are now making, this is primary antibody, assume that we're calling primary antibody as a, because we are using it first. This is an about applied, nothing, don't mix it up with immunology. These are immunocomponents nevertheless. So secondary antibody we are raising against the primary antibody, constant region of it. It will recognize somewhere this part like this. Okay, I hope you understood the critical points on Western blotting. They're usually, everything is simple. Everybody will say you run the electrophoresis, then you do the blotting, then you add primary antibodies, secondary antibody. We have to, if we remember these two things, that is blocking, that is one part, and why should we use secondary antibody and not just primary antibody directly? These 2 points, if you remember, you know some critical details about it. And what can Western blotting say and what it cannot say? Western blotting can tell if there is a protein present or not. It cannot tell if the mRNA of that protein is present or not. It cannot tell if the gene for that mRNA is present or not. If there is a protein, the mRNA should have been there. If the mRNA is there, the gene should have been there. That's easy. But if you see a situation like this where there is no band, you cannot say that there is no mRNA, right? You cannot say that. If or you cannot say anything about whether the gene is there or not. If the band is present, gene and mRNA are present. If the band is absent, you cannot infer anything out of it. You can only infer that the protein of interest is not present in that sample. That is what you can say. Okay, and every time you are doing an experiment, I think I will go to the next one later. I will describe what I wanted to. So I hope you know 2D gel electrophoresis. I think I want you to know what are ampholites. You should know about PI. If you're in 4th year, and next year you will be going for interviews. If you're interested in science, or you might be facing a lot of interviews. If you're facing, if you're prepared, you want a quick preparation for interviews or quick preparation for any of the competitive examinations, including GATE and CSCR, what one should do is read the first 10 lessons of biochemistry. Okay, you can choose any of the standard books that are there available. Try to read those very, very carefully and clearly, including the experiments that are given in there. My favorite book is at that time probably Strayer. Okay, you might feel it little complicated, but once I don't think it is complicated. I don't know why people don't take it up, but Strayer is also good. Leninger is also good. White and white, anything is fine. The first lessons are the fundamentals and foundations for the rest of cell biology, molecular biology, and everything else. If we don't know how molecules behave, right? Biochemistry or chemistry itself teaches about how molecules behave. And when you have 3 different molecules, what might happen? If you have 4 different molecules, what might happen? And biochemistry is about molecules, biomolecules, right? And you also have something like enzymes. So anywhere you are, you have to study these as a preparation for interviews or for examinations. If we are not clear on these, and whatever higher things you build upon these are, it's not going to be like a memory. It's like it'll be short-term memory probably. So if we should know about amino acids, we should know about proteins, about the charges and how the structures actually change, and then the isoelectric point, what determines isoelectric point, acids and bases. Many other similar ones should be dealt. Okay, I'll take this in the next class. But if you have any questions, you can please tell me. You can ask me now. And I also would like to ask you people, I want a list of topics that you want to you want me to repeat, because I want to do that before we go into the papers. In papers, it's going to be at a research level. We are not talking as a learning thing. We are talking like it's not like we don't learn it as students. We should learn it as an expert. So before we move on to that level, I would want that you give me, don't make it, everything cannot be repeated. Actually, if you have noticed, I have repeated many techniques at least twice. Okay. So if you make a list, and then I will try to revise some of the topics in one of the class, but you have to tell me in advance so that I have to prepare the PPT and think of how differently I can present it. Okay. With regard the assignments, this is the last point I will tell today before I close this lecture. What I want you to remember is in the assignment, although there are a few mistakes, I overlooked them. Okay.